Longevity Junky is presented to you by our friends at 8cast. Hello, Longevity Junky. It's time to get your fix. I'm Nikki Lee, the original OG Longevity Junky.
And I'm Buck Joffrey, the health-conscious hedonist.
Coming up, the role of AI in advancing medicine. And Buck talks with Dr. Joshua Hoffman at Galleri, an amazing new test that screens you for up to 50 types of cancer. So, Buck, how you been?
Well, I'm doing all right.
Standing up straight, bubble vision fix. What's going on?
I'm standing up straight, and I've still got the bubble in my eye, and I can sort of see. I can see over the bubble. I had this emergency retinal surgery a few weeks back, and
you had to stare at the floor for 2 weeks.
I did stare at the floor for a couple of weeks. That's fine. But now I'm back in action. My real estate group is buying a bunch of real estate right now because it's a really good time to buy real estate.
Because it's down, the market's down, or the interest rates are down?
Well, what happened was interest rates went up really fast, as you know, and it killed people. It killed, And so in these kind of situations, there's two ends to this, right? So if people have to sell because they're in distress, then there's buyers, and I'm on the buying side right now. So this is when I think, Warren Buffett talks about how you should be greedy when others are afraid. Right now, people are afraid and we're being greedy. And that's what's going on with my investor group. So I'm trying to ignore the eye bubble and focusing on the acquisition. So yeah, that's the excitement. But I'm sure you have something far more exciting than that.
Well, I went to Norway
there you go.
Gosh. Yeah, I went to Norway. I went for the royal wedding. I was a guest of the Princess of Norway and her now husband, Shaman Durek.
I didn't know they were a monarchy.
I know there's there's a lot more princesses out there than we know about.
Yeah. I mean, there was the one what was the movie with
Princess
Let
Diana?
It Go.
Oh, Frozen? Oh my God. You can tell you have 3 little girls.
I do.
But you know what? Honestly, it was so beautiful and so majestic. And 2 or 3 days that we were there for the wedding, it was all sunshine and blue skies. I mean, It was the most absolutely beautiful wedding I had ever been to. Such love in the air. So much paparazzi. People camped out. It was incredible to see so much energy there. And every morning I got up super early because the festivities started pretty early as well. And I would go and swim in the fjords and write in my gratitude journal on a dock. And oh my God, it was like the best thing in the whole wide world.
Maybe I can be your +1 next time you go to a royal wedding.
Yes, next time for sure.
I'm gonna try to marry into a monarchy if possible. Well, I'm
trying to find my prince. Exactly.
We don't have to sit next to each other. I'd be working up my own princesses over there. Come on. Minnesota is full of Norwegians, right? Where I grew up. At least it used to be that way when I was a kid. It's changed a lot, but and apparently the Norwegians moved there because Minnesota looks a lot like Norway. I didn't know that.
I've only been to one part of Minnesota.
Yeah.
So I feel like I would have to explore more. But if that's truly the case where it looks like, I hope the air quality is just like Norway too, because that's one of the things. Yeah, it's clean.
It used to be. Yeah. I mean, I think it is. I haven't lived there in years and years and years. I just go back for Vikings games and seeing my parents. That's about it.
Honestly, every time I'm in Norway, I just keep saying to myself, this is where I want to be. I want to live here, or at least have like a house there, something, because it's just my nervous system. I legitimately feel my nervous system just get so relaxed.
Do you still have that Vikings outfit that you wore the first time?
No.
No,
that was a legit Viking garb, and it was from there, and so I left it there. And I don't know if I'd ever wear it again except for Halloween. So anyways, but
anyway, okay.
So let's go on to the need to know segment. You all need to know about the role of AI in advancing medicine. I mean, I know we all know about it with Chat GPT and things like that, but to hear that AI now is in advancing medicine I'm very intrigued on this. So, Buck, what's the latest?
Yeah. I mean, when you think about artificial intelligence, right, a lot of people are a little bit terrified and for good reason.
Well, people are afraid of losing their job.
They're also afraid that the robots are the computers are going to take over and kill us, right? Have you seen The Matrix? I mean,
yes. But I think of another movie. I think of I don't know, is it like iRobot or something like that? There's another robot movie where that was the one that freaked me out. And I don't know if it's iRobot or not.
Well, here's the deal, right? So they might, the artificial intelligence to the computers, eventually maybe they will kill us off. But in the meantime, they're going to help us live longer. And the way they're going to do that, there's lots of examples of it already. So you've got, for example, drug discovery, right? Now, finding new medicines used to be a super, super long and expensive process. And we're talking about probably a decade or two, billions of dollars just to get one drug from the lab to the pharmacy. But what artificial intelligence is doing is it's changing the game. So it's like, imagine having a computer program that can read through millions of scientific papers, scan thousands of chemical compounds, and actually identify sort of patterns. That's what AI, one of the things that AI is really good is find patterns that even the best scientists are going to miss and say, gosh, maybe this will be a good medication based on this. So instead of sifting through that in a human sort of slow way, you're doing this using artificial intelligence. So,
well, and AI learns faster. I mean, it learns about instantly rather than humans have to go and study and, Yeah,
it's going to look at tons of data to find out which molecules could work as medicines. Like shifting through, it's sort of like sifting through a giant puzzle, right? Where all the pieces are possible molecules that could help treat a disease. And AI sorts out through all these puzzle pieces really fast, figuring out which ones might fit the disease puzzle and solve it. So that's that. And then there's an example of this, actually. There's a company called Silico Medicine that used AI to find a possible treatment for a lung disease. And it only took them about 46 days to do this. And without AI, it would have taken, like I said, a decade or two.
Wow.
The other area of AI I think that's really interesting is vaccine development. Okay. Now, so let's talk about vaccines. AI is playing a huge role here. And traditionally, making a vaccine could, again, take a decade or more.
Don't tell people of COVID that.
Yeah. Well, that's what I was going to say, because with the COVID thing, AI was a big reason there was an effective.
You think they used AI? Because they didn't mention any of that.
Oh, they did. They absolutely did. It was one of the big reasons they were able to find an effective vaccine target. So how did they do it? They analyzed the virus's genetic makeup. And what they did is, so think of the virus as having a blueprint, right? And the AI scans this blueprint and finds its weak spots. And so for COVID, the weak spot was what is called the spike protein. You probably heard of the spike protein. So AI identified that really quickly. That's the part that helps the virus actually get into our cells. So AI identified that target and help the scientists ultimately focus their efforts on creating a vaccine that would trigger the immune system to recognize and attack this spike protein. And then it also sort of optimized the vaccine. It helped figure out the best way to design, in this case, the messenger RNA that would be stable and effective once it entered the body. So ultimately, it's like it kind of did all of this stuff that would have taken years and years to do in traditional ways by just putting it through this massive amount of, data crunching that can happen within seconds. I mean, you've seen chat. It is incredible what you can do and how quickly you can do this.
Yeah.
Now, we talk about vaccines and people just immediately think of COVID and all that. But listen, this is a very, very big area. So maybe we can find a vaccine, and there's already some work on vaccines against cancer, right?
Sure.
So there is actually some work on this already.
Right.
But what I would say is that, I mean, for me personally, if there was a vaccine that we believe could prevent various types of cancer, I would do it in a minute, because as you'll see in our interview coming up, early prevention or early detection is really our best shot at beating cancer.
Oh, yeah.
So in the case of vaccines, whether you want to do this kind of stuff or not, but there is plenty of research coming down the pipe that's going to potentially help us to use vaccines in ways we don't think about. I mean, normally we only think about it in terms of viruses and that kind of thing. But if you can inoculate against disease processes like cancer, then, I mean, there's a whole new world that is potentially available to us. Now, it's not just about drugs and vaccines, but that's just two examples of how artificial intelligence is going to help. One of the things that I think of, which I think is going to really speed up the whole longevity escape velocity issue, which basically means our ability to outrun the diseases that kill us, is going to probably come from artificial intelligence. Because, we'll talk more and more about drugs and supplements, but how our ability to identify those things that can potentially really help us live longer has just gone up by millions fold using artificial intelligence. So I think it's a really, really exciting area. And we're going to see how, in the interview that we do today, there is a use of AI. And you can, we'll talk a little bit about that again afterwards.
Nice. Okay. Well, we're going to take a quick break. And when we come back, we'll talk with Dr. Joshua Hoffman about a groundbreaking new AI cancer test called Galleri. Welcome back to Longevity Junky. Time for our guest interview. Bob spoke with Dr. Joshua Hoffman in The Breakdown.
Josh, welcome to the show.
It's my pleasure to be here. Thanks for having me.
Well, we're going to talk in a fair amount of detail about this test that Grill has called Galleri, which is really significant potentially in terms of improving human longevity and health span and all that kind of thing. But before we get into that, the test is related to cancer, and it's related to detecting early cancer. I thought it would be useful for people who really don't know that much about the biology here, is just to give a little bit of relevant background in terms of cancer biology, so we're on the same page. Obviously, you and I are both physicians, but let's start with some of the basics. Like, what exactly is cancer in the first place?
So cancer really is a disease of the genome. It really is very much related to the genetics and the heritability of your DNA. But it is really uncontrolled cellular growth. And there's a number of reasons, many, many different reasons why cells will grow in an uncontrolled way that become cancerous. But generally, in many of them, they're related to the genomics. Now, what we've learned about genomics is it's not just the code of the genome, right? So your DNA is made of a code of 4 nucleotides. But what we're learning now is that there are molecules that attach to the DNA that regulate the genetic code, and they turn genes on and off.
And that's referred often as epigenetics, right?
Exactly.
People talk about the genetics being the part that maybe codes the actual, protein itself, and then you have the epigenetics, which is the material around that, around those sequences, which before, frankly, we didn't think much of, right? And now we're figuring out that they actually do mean something.
But that's exactly right. And so when we say, interrogating the genome, learning about the genome, it's a disease of the genome. That includes the code of the genome and all the things that are attached to the genome, like, these molecules that we call methylation molecules or methyl groups that attach to the genome that turn tumor promoter genes on, they turn tumor suppressor genes off, and they do that across many, many different kinds of cancer. And so they're very related to cancer and the development of cancer.
Right. And so one of the things I want to kind of, and this will lead into the test later, but each one of these potential cancers and the mix of the genomic sequences, the mutations there's, and the epigenetics, these methylation points in the epigenome, they lead to essentially various types of fingerprints, right? And these fingerprints are basically kind of what we're looking at when we start looking at potential early detection. Is that correct?
That's right. So most genetic tests up till recently have focused on mutations, right? changes in the code of the DNA. And many of them are associated with cancer. But again, for people like myself who are over the age of 50, I have many mutations in my genome that are similar and the same as cancer patients have, but I don't have cancer. So these cancer mutations are uncommon. Some of them are very important for cancer, but some of them are also shared by people who don't have cancer. So they're not always, they're important to know about if you're diagnosed with cancer. Do you carry this mutation or that mutation? Because it can tell you a lot about the cancer and its potential treatments, but they're not great ways to find cancer because they're uncommon and they often also exist in people without cancer.
One of the interesting things that I think people don't know, and maybe you can comment on this a little bit since you're in this space, is that cancer cells, it's not uncommon for cancer cells to exist within healthy human beings, but we have something called the immune system, obviously, that's constantly surveilling our bodies, right? And if you want to talk a little bit about, how that function works, that might be useful as well, because there is this idea that maybe you have cancer cells, but you don't have cancer. So what's the difference there?
Well, it's a great point. And we're on the early innings of really understanding that interaction. We do know that our immune system is surveying the body all the time and does have the ability to kind of shut down early cancerous growths. The extent to which that happens, where it happens, when it happens, and why it happens is something that we're still beginning to understand. But we do know that the immune system plays a really important role in what we call immune surveillance of cancer. But we also know that our prior understanding of cancer is less than complete. And we are now, by looking at the biology of cancer, like the DNA from cancer cells that we see in the blood, the immune signatures that we see in the blood, we're learning all kinds of new things about cancer now. For example, the old way that we used to stage cancer is pretty crude relative to looking at the biology of cancer by looking at its DNA, and those signatures are much more sophisticated. And as a result, we're learning different things about the natural history of cancer.
Just give us some statistics on this. Like, what percentage of people develop cancer in their lives?
Somewhere between 40 and 50% of men and women will develop a cancer in their lifetime. It's very high. We know that cancer is soon to become the number one killer of men and women worldwide. Right now, it's ranked number 2, but I think the epidemiology suggests it's going to overtake cardiovascular disease very shortly. And why is that? Why is it such a killer? It's because we're finding most cancer too late. When it's already spread, symptoms are present, and there are very few treatment options. So we know one of the only ways to really begin to, we've been fighting this war against cancer, as you know, for 50 years, and it's not one we're winning. So we need to open up kind of a new front in the war on cancer and get much better at early detection when cancers can be cured.
As it stands, there's various guidelines for early detection of certain cancers. Which cancers are those? And, tell us how effective those are right now.
We screen for 3 cancers in women: breast cancer, cervical cancer, colorectal cancer. We screen for 1 or 2 in men: colorectal and prostate. And then for heavy smokers, whether you're a man or woman, we have a low-dose CT for screening for lung cancer in heavy smokers who meet that criteria. So all told, if everybody, if you look at adults over the age of 50 who are at elevated risk for cancer, those 5 screening tests are finding about 15% of the incident cancers in that population. So you can see very quickly, that's not going to do the trick.
Yeah.
We're not going to shift the cancer mortality curve or bend the cancer mortality curve or win the war on cancer, finding 15% of the cancers at an earlier stage. When I went to get my colonoscopy when I turned 50, I felt pretty good about myself. What I forgot, what is that I was 10 times more likely in that very moment to be diagnosed with some cancer I wasn't even looking for. So we live in a world of single cancer screening right now. We screen for one cancer at a time. We look at shadows and images, or we directly inspect tissues. And though that's quite limited, right? So you're only going to find those very few cancers, and you're much more likely to be diagnosed with one of the many cancers that we're not screening for than those. And then those are also limited because they have very high false positive rates. And many of the cancers, like early encapsulated prostate cancers or the early hormone positive breast cancers, people may be more likely to die with than die from. So those are the limitations of current screening.
The cancer deaths that occur on a yearly basis, 70% of those cancer deaths are occurring in cancers that we're not screening for at all, that we're just basically playing defense on. Is that right?
That's exactly right. So many of the deadliest cancers, stomach cancer, esophageal cancer, pancreas cancer, ovarian cancer, uterine cancer, we have no recommended screening tests for at all, liver cancer. And so it's kind of the classic streetlight problem where, you drop your keys and there's a streetlight over here. So that's where you're looking. Yeah. But all the deaths are happening over here in the dark.
We're going to talk a little bit about Galleri now, which is the test. The biology behind the blood test that Grill is doing kind of has an interesting backstory. It sounds like it was involved in trying to sort of minimize the use of amniocentesis. You want to tell us a little bit about the backstory there? Because I think it's pretty interesting.
Thank you for asking. It's actually a fascinating story. So at Illumina, which is the biggest sequencing company in the world, the chief medical officer there was someone named Rick Klausner from the NCI. And they had a hypothesis that, because the mother and the fetus are sharing blood through the placenta, that we might be able to look at the mother's blood to determine whether there was a chromosomal abnormality in the fetus, rather than sticking a large bore needle into the uterus in the amniotic sac and drawing out the fluid, because that carries a risk of aborting the fetus.
Right.
And so they did a study in 125,000 women, healthy, pregnant women. And sure enough, they learned that, yes, we can find the fetal chromosomes in the mother's blood. But there were 10 really abnormal cases where they looked at the DNA of these women, and one of the lab directors, a woman, and Dr. Klausner said, you only see abnormal DNA like this in people with cancer. Bring all those people back. And those 10 women all were found to have a cancer, many different types of cancer. And that was when the light bulbs went off within Illumina saying, we may have just found a way to find asymptomatic early cancer in people who have no idea they have cancer by looking at the DNA circulating in their blood. So they spun out Grail as a private company, and Grail raised a lot of capital to do some of the largest studies ever done to figure this biology out and developed this asset.
And that kind of brings us to what exactly sort of the technology is, because it's based on this concept of cell-free DNA. Talk about what cell-free DNA is. I mean, it's kind of what it sounds like, but why is it of significance?
Well, it's really important. So just so that everybody understands, all cells in our body carry DNA. And when they die or are injured, they release that DNA into your blood, as long as there's access to the bloodstream. And so there's a lot of DNA that's circulating in our blood, and we call that cell-free DNA. Now, tumors, by definition, cancerous cells, are growing faster than our normal cells most of the time. And so when they die, they also shed their DNA into blood. So when you get a sample of DNA in someone's blood, it's a mixture of healthy cell DNA and tumor DNA. And Grail, when it was founded, did a very unbiased and open experiment. And they said, let's look at all this DNA in the blood and let's test all the available methods about how to read this DNA. We'll look at mutations. We'll look at chromosomal changes. We'll look at RNA. We'll look at proteins. We'll look at DNA fragment lengths. And we'll look at the methylation status of these DNA fragments. In other words, how are they methylated? Are the genes hypermethylated? Are they hypomethylated? And because there are very consistent patterns of methylation in cancer. These are all the hallmarks of cancer. And they compared all of these approaches head to head. And they learned that by far and away, the best way to detect cancer was by looking at these methylation patterns in these fragments of DNA.
Yeah, again, we've I think a nice way to think about it for people, again, to me, is like methylation patterns. We talk about epigenomes. So really what we're talking about is, these DNA-related fingerprints of cancer, that we're using all of this information that we have to put together and say that when there's cancer in the body, there's these various fingerprints in the blood that you're able to pick up on. Is that a fair assessment?
That's exactly right. And that's why machine learning has been used, because we've taught these machine learning algorithms by doing these very large studies to recognize these very common patterns, these fingerprints that you only see in cancer. You almost never see these types of fingerprints in people who are known not to have cancer. And the other important thing just to mention, these methylation groups, these epigenetic patterns, they also carry another fingerprint, which is where in the body, what organ or tissue that signal comes from. So our Grail test called Galleri is a two-step test. So you sequence the DNA for these patterns, you subject it to a machine learning classifier that says that's a cancer signal or not. And then if it's a cancer signal, it goes through a second step that says we predict that cancer signal is coming from the ovary or the pancreas or the stomach. And we do that with very high accuracy.
I'm just curious, when you're using machine learning, you're using artificial intelligence. Obviously, medicine is one of the places where we're probably going to see the greatest impact of artificial intelligence. Does that mean that the test that you have, the Galleri test, is actually improving in real time, sort of because you're getting constant feedback?
That's right. So there's a certain way you have to do it. You can't just do it on an ongoing continuous basis because you need to revalidate and retrain. So yes, that's exactly right. Because Galleri is now out there available as a lab developed test in the world, we've now done over 100,000 tests. We're using that data now to train the next version of our algorithm. And then we'll revalidate that. And then that'll be the next version of Galleri. And it will improve its performance by now having seen a much broader array of different cancers and different signals and in a lot more people.
How many cancers are we talking about right now that Galleri is able to detect?
Well, there are hundreds of types of cancer, and we're still grouping them. For example, we call, there are 4 or 5 different types of breast cancer. We call breast cancer 1. There are many kinds of lung cancer. We call lung cancer 1. So we're still doing a lot of grouping, but if you ungrouped everything, even a little bit, we're absolutely, with all the grouping, we're over 50 different types of cancer. If you ungrouped them, obviously it would be a much bigger number than that.
Right. Well, and that's obviously a big improvement compared to the 5 that you mentioned.
That's right.
Earlier.
Remember, again, as you mentioned, most of the cancer deaths are occurring from all these other cancers that we're not looking for at all.
Not that it's like a head-to-head battle, but I am curious, like if you take like a breast cancer, typical, the type of screening that we do today, what kind of, sensitivity that is compared to, the type of testing that you're doing with Galleri. Like if you go head-to-head, do you have statistics to say one's more likely to find that cancer than the other?
Well, we know quite a lot. I mean, again, it's really important for everybody to understand that the Galleri test is not intended to replace existing single cancer screening tests. It's proposed to be a complement, kind of an adjunct test. Add to it. Because remember, these tests were designed very differently. So mammography is a great example. Mammography is looking at shadows from images taken of the breast tissue, and it's trying to find everything. It's trying not to miss anything. And in fact, of 100 women who have a positive mammogram, only about 5 to 6 of them will end up having breast cancer. So it surveys everything very broadly. Now, Galleri, because it's looking for a very common signal that's shared by over 50 or more cancers, we've done the opposite. We're trying to be very specific. In other words, they're focused on being very sensitive, and their positive predictive value, which means if you have a positive test, how often is it cancer? Their number is about 5%. Our numbers are the opposite. So with Galleri, we will miss a lot of the breast cancers that mammography may find. But if we find a signal for breast cancer, the likelihood that it's going to be cancer is somewhere between 40 and 50%. Much more, an order of magnitude better, because it's so much more specific for cancer. But we know that a lot of these slow-growing cancers, like slow-growing breast and prostate, that are found by imaging, and lung. Again, these nodules in the lung that low-dose CT finds are often not cancer, and they're not going to kill people. They're also not shedding a lot of DNA into the blood. So Galleri is not going to find those cancers that we worry about that we're overdiagnosing today.
When we talk about in medicine, we talk generally about these terms that you're using, like sensitivity, positive predictive value. We talk about specificity, but for the test as a whole, what kind of? Let's talk about positive predictive value. I think you mentioned it already, but let's define that and kind of give us again a sense of what that is with this test.
So again, it's the most important metrics for a multi-cancer early detection test are going to be different than the metrics for a single cancer screening test, right? The single cancer screening test, there's just one organ that they're looking at, and they want to be very, very sensitive. They don't want to miss anything, and they accept very high rates of false positives. So, stool-based colon cancer screening, low-dose CT, mammography, the false positive rates are well over 10%.
Sure.
Right. And we accept that because it's just a single organ and we know what those next tests are. For a multi-cancer early detection test, you can never accept that kind of false positive rate. So our false positive rate for Galleri is 0.5%. It's 0.5%, an order of magnitude lower. Now, what does that mean? That means that if you don't have cancer, we are very unlikely to have a positive result. Now, because cancer is very, it's only about 1 to 1.5% of the population has cancer. The positive predictive value is a very important measure as well, which is if you have a positive result, what's the likelihood that you have cancer? And again, for single cancer screening tests, that number is somewhere between 5 and 10%. Okay. For Galleri, that number is somewhere between 40 and 50%. So an order of magnitude better. So when, if you're a doctor and you're sitting in front of a patient who has a positive Galleri test and a predicted location of the esophagus, that person almost has a 1 in 2 chance that they're sitting there with cancer right now. And it's serious business. You got to go look for that cancer because you're likely, to find it.
Yeah. So is there variability though, in terms of the positive predictive values per different type of cancer within the Galleri test?
So the positive predictive value is really associated with that signal, that shared cancer signal. Okay. But you're right. The sensitivity is different for every different type of cancer because the sensitivity is related to how much DNA is being shed by those tumors. So let me give you an example. Pancreatic cancers. The deadly, fast-growing invasive cancers are shedding a lot more DNA, even in their earlier stages, than slow-growing encapsulated prostate cancer or slow-growing thyroid cancers or slow-growing breast cancers. So we're going to have much higher sensitivity. So we've done an analysis where we looked at a pre-specified subgroup of 12 of the deadliest cancers. And our sensitivity for those deadly cancers in stage 2, which is still curable, is 70%.
Wow.
Right?
So that's remarkable, especially because you're talking about pancreatic cancer here, which, I mean, most people who are, in their 40s and 50s have heard about a story about somebody getting pancreatic cancer who was totally healthy and then they died 3 months later. I mean, that, I was even talking to a surgical oncologist friend of mine yesterday, and he wanted me to ask you about that specifically is, how is this going to benefit us there? Because that's what he's getting, that's his worst outcome patient.
That's right. Most pancreatic cancers are found after patients either have signs or symptoms. And when that occurs, it's usually too late. That means that cancer has already invaded into the biliary system or invaded the nervous system and has already spread beyond the pancreas. And so we already, in our studies and in the real world, have found multiple stage 1 and stage 2 pancreatic cancers. And the surgeons say they've never seen this before. And they tell their patients that it's like they hit the lottery,
right?
Because they took the Galleri test and we found these.
So, there's also some cancers that are probably not going to be as good for Galleri. I mean, in a former life, I was a neurosurgery guy, and you have the blood-brain barrier. Those types of issues may confound some of those findings or make it more difficult to, to find brain tumors, things like that. Is that right? And maybe talk about some of the limitations.
Yeah, that's absolutely right, Buck. And so, for example, we're very clear when we educate doctors about our test that we're not going to find brain cancers because most brain cancers are protected from the bloodstream. And so as they shed DNA, that DNA from those cancers is not going to get into the blood. It's going to get into the cerebrospinal fluid. And so we're not going to be able to find brain cancers. There are other very slow growing cancers like thyroid, some of the prostate, some of the breast that we're not going to find well. We will find the aggressive triple negative breast cancers very well. We'll find the aggressive prostate cancers even in early stages well. But the slow growing, more indolent phenotype cancers that people are worried about that we're overdiagnosing, we are not going to find those well. Some of the very slow growing urothelial cancers, the DNA is much more prevalent in the urine than in the blood. We find those, but not as well as some of the other ones.
Yeah. I'm curious too, I obviously, because I think about the brain stuff a fair amount, but is there any thought on screening? I mean, the problem, I guess, is you don't really want to do a spinal tap once a year to test CSF and that kind of thing, right? I mean, any other potential solutions around that you can foresee?
There are, and we're thinking about them, and other companies are as well. There's some CSF that interacts with your sinuses and can get into your saliva. So there may be other biofluids that can help us, because you're right, getting cerebrospinal fluid from a spinal tap is not something that's going to be easy to do as a population screen.
So typically the idea with a test like this is maybe, is your vision that maybe you would do it once a year in addition to whatever other screening you're doing. Maybe you even started earlier than some of the other screening because it's pretty easy to do.
So based on everything we know about the biology and the natural history of cancer, and everything we know about DNA being shed from these tumors and how that evolves, we believe that about a 1-year testing interval is about right. Now, we may learn as we learn more that needs to be every 6 or 9 months. But right now, we think a year is a good way if you're trying to optimize the number of cancers you find in the population. Because what we're learning by looking at the DNA in the blood is that these aggressive cancers are moving pretty quickly, which is different than what we often saw with staging. when you're looking at imaging or feeling the cancer in surgery. So the movement between stage to stage is a little different than what we're seeing with the DNA. And so I think we're rewriting the textbooks as we're developing this assay. But you're exactly right that, in annual screening interval is what we're recommending right now. And in about a year or two, we're going to have data from studies where we've done several consecutive annual tests in individuals. And we will get even more information because right now, all the performance, as good as it is just based on a one-time test in an individual. We haven't even begun to layer in the data within an individual, how their signal is changing over time. That may be much more informative than what your signal is at any given point in time.
So my friend, who Alan Viglione, Dr. Viglione, he does some of these shows with me, and he's here in Santa Barbara, and he's the one who suggested that we touch base. And one of the things he mentioned to me is that he saw you talk, and there was a bunch of oncologists in the audience that really were not interested. They were not happy about the existence of this test. And frankly, when I hear all of the potential behind this test, it really surprises me. I mean, to me, this is like a very exciting potential way of saving thousands of lives. So why would the oncologists be totally against this?
Well, it's not, there may be some oncologists that are not on board yet. It's very disruptive and very new. So we spend a lot of time with oncologists. Some of them have been founders and advisors to GRAIL from the beginning. And the oncology community largely has really embraced what we're doing. But remember, oncologists are not the ones who usually screen for cancer.
Right.
And so they're not as familiar with all the issues around cancer screening. In fact, when I ask oncologists sometimes, what is the positive predictive value of mammography, they say things like 30, 40, 50%, which is not at all correct, right? Because that's not, they're used to treating cancer. And, and again, we think that if we can find cancers before symptoms present in their asymptomatic state and in earlier stages, there are many more treatment options, many of which are surgical and radiation and not drugs. And so that's also a difference. But we know drugs are moving earlier and earlier across many cancers. So there's a lot of excitement in the oncology community, but there are skeptics. And the skeptics...
So what are the skeptics? What are they worried about? Like the ones who are worried about it?
They're worried about, how do we know that it's good to treat all these early cancers? Well, we know generally that for solid cancers, we've got enormous amounts of evidence for breast, lung, colorectal, others that when cancers are treated early, they have better outcome. Even in the same stage, if you have an asymptomatic stage 3 and a symptomatic stage 3, the asymptomatic stage 3 generally has better outcome. So we know that, but it has not been clearly and definitively established for every cancer type because most of these cancer types are extremely rare. They're uncommon.
So, obviously this is going into the hands of primary care docs. And part of the issue is they need to be trained on this stuff, right? I mean, if they get positive results and you send somebody for imaging, and if it's negative, what do you do? I mean, I don't even know what you would do in that situation. Maybe you can comment it, but maybe you could talk about, some of the training that's involved. And also, as a follow-up to that, what do these people do if there's, they can't find a cancer and these people are screening?
Well, let's go to this. So first of all, we have very extensive educational training that we do with doctors. And if a cancer signal, if you ordered a test, for example, and you got a cancer signal detected result, one of our medical science liaisons who are in the field would be in contact with you within 12 hours about that individual case and seeing what information you needed. Do you need any resources? How can we help? We have experts at the ready to talk to you if it's a complicated case. So we have a huge amount of resource available to support the doctor if they get a signal positive case. Now, when you do get a signal positive case, let's just say that case, uterus, positive signal uterus, you do a pelvic CT and it's negative. Your question is, okay, now what do I do? Well, you've got a couple options. One, it could be that the lesion is too small and it's just not imageable. Imaging is not perfect. So you might bring the patient back in 3 or 6 months for a second image. You might just consider it a false positive right then and say, you know what, it's probably a false positive. We'll do Galleri again next year and we'll chalk it up to that. Grail also offers a free repeat test in those cases, and you might just repeat the Galleri test because sometimes it might be positive again, in which case you'd really want to investigate further. And then you want to talk to the patient to see maybe the signal is actually a cancer signal, but it's not in the uterus. It's in the spleen, or it's in the liver, or it's in the stomach, and we need to look somewhere else. So are there any other clues about where else to go look? And then every once in a while, someone will say, I feel very strongly that you may have cancer right now. Let's do a whole body MRI or a whole body image of some kind. But that happens pretty infrequently.
So tell us about any data that you have right now, or maybe some of the studies that are happening with regard to using Galleri in the clinical setting. Maybe when you data to support what happens when you combine it with some of the standard of care screening that you talked about.
The first study we did was called the Circulating Cell-Free Genome Atlas, which was a group of 10,000 patients with diagnosed cancer, newly diagnosed, and 5,000 adults without cancer. And that enabled us to really develop the test, validate the test, get to a new version, test it, and validate it again. Then what we did is a real-world interventional study called Pathfinder. This was a seminal study in the field. It was 6,600 adults over the age of 60 from 6 or 7 centers around the country, like Mayo Clinic and Cleveland Clinic, Sutter Healthcare, Oregon Healthcare, and we had very special centers. And these were asymptomatic individuals. And what we did is we added Galleri to their standard of care screening. And we followed them all for a year. And what happened? Well, adding Galleri doubled the number of cancers that were found in this population on top of standard of care screening. And half of the cancers that were found by Galleri were in stages 1 and 2. So again, really remarkable finding in a real world population. It was safe. There were no adverse events that were significant. The satisfaction of the people who took it was high. Even when there were false positives, their satisfaction was very high. They were glad they did the test. There were false negatives. There were false positives, as you would expect. But we doubled the number of cancers found compared to standard of care screening, which is very consistent with some of our modeling that we've done and published. There's also a large randomized clinical trial going on right now in the UK, in the National Health Service, of 140,000 adults over the age of 50 randomized to standard of care versus standard of care plus Galleri. And we're already almost done with the second year of that study now. And so that will be a huge study. We're also following all the tests that are being done in the real world, over 100,000. And we're seeing findings that are very similar to what we've seen in our trials, which gives us a lot of reassurance and confidence that the test is performing well.
Do you have any sense, I'm sure you guys have looked into this, that if you get sort of accepted into the standard of care of screening, even just say in the United States alone, like what is the potential in terms of lives saved?
So we've estimated that, and this has all been published based on models, pretty conservative estimates. But, as I mentioned today, if you just look at adults over the age of 50, there's, well over 1 million cancers that are diagnosed every year. And if you added Galleri to our standard of care screening, instead of only finding 15% of the cancers by screening, you could now find 50% of the cancers by screening and 75% of the deadly cancers you could now find by screening. So if you add that up and you say, okay, what is the impact on life expectancy or the number of deaths averted, that calculates out to about 100,000 deaths averted every year, which is about a 39% reduction in the expected deaths over the next 5 years. That is about the same magnitude as everything that we're doing in cancer, everything. If you add up everything we do in cancer, screening, diagnosis, treatment, hospice, surgery, everything, we're averting about 100,000 deaths a year. And you could have that same magnitude of benefit if we just adopted Galleri and everybody took it based on the models.
Which is, of course, now, like, anybody listening to this wants to probably, think about getting Galleri tests. It's not something that's currently covered by insurance.
There are some who cover it, not many. There are many self-insured employers who are providing it to their employees and their payers. So we do have some coverage. There are also some life insurance companies that are providing it as a benefit to their care, their covered lives. But we are in an accelerated approval breakthrough designation with the FDA, and we're hoping to get FDA approval within a few years now. And then we hope to get Medicare coverage as well. And we think once we get the FDA approval, that many of the payers will come along. Right now, we have New York State Department of Health approval, CAP-CLIA certification. So we're available out there, but it's most people are paying out of pocket right now if they don't have payers coverage.
How much does it cost typically right now?
So typically, if you're just walking in off the street to your doctor, it'll cost about $949. But if you're part of large group practices or large concierge practices or other health systems that we have partnerships with, there are discounted rates that are available for them.
Dr. Hoffman, Josh, thank you so much. It's been just a really incredible technology and the best of luck with it.
Well, thank you so much. Pleasure to be here.
We're going to take a quick break and we'll be right back with our healthy takeout. Welcome back, Longevity Junkie. Time for Healthy Takeout. Wow, Buck, what an amazing test. It's truly a game changer for our approach to cancer.
Yeah, well, let me ask you. I mean, obviously, I was the one who got to do this interview. I did lucky. A while ago. What are your thoughts, takeaways?
I had a lot of surprise. Like, there was a lot of information that hit me that I had no idea. Like, first of all, that we only have 5 tests for cancer, and there's like 50 types of cancers that it is still not detecting, right? Like those 5 specific tests only count for, like a fraction, a real minor fraction. Like, what was it? Like 10, 13% or something of all cancers?
Yeah, basically the ones that we don't have, the ones that we have screening tests for make up very, very few percentage of the number of cancers that are out there. So,
right.
So the idea is that, So
I had no idea.
Yeah. I mean, it's crazy because, there's lots and lots of cancers, but there's only a few that we can actually screen for right now. And that's where this thing really makes a big difference.
Yeah.
But, let me kind of talk a little bit because I think it was sort of complicated, but I'm glad you brought that question up because, again, this is where artificial intelligence, I know it is being vilified across multiple industries. I know Tim, for example, was talking about how in Hollywood, this is a major concern. People are concerned that it's going to take away our jobs and so on and so forth. But there's a lot of positive things that are coming out of this as well. So let me explain kind of how this works. So in general, the idea, just in summary of this, is that when you have cancer cells, even in very early stages, you can essentially get these little pieces of DNA in the bloodstream that are cancer-oriented, are from cancer cells. And what this test allows us to do is to take blood and see little patterns of DNA fragments and use artificial intelligence to look at those patterns like fingerprints. And it's almost like looking for a suspect. Because if you see these various fingerprints when you're going through that artificial intelligence, then you might say, oh my gosh, this looks like lung cancer. And we can't see it so much, or maybe we can't even see it on an x-ray yet, but this looks like lung cancer, early lung cancer. It's like the fingerprints are left behind. And that's why it becomes really, really powerful. Does that make sense?
Totally. And when you say that, like, it might not be able to be shown in like a mammogram or an x-ray, I feel like that is, if you can see it, then it's probably too late, or it's already really developing in the system, because we're talking about like in blood, like very microscopic detection. From what I've gathered in the interview, that it's kind of too late almost. Like that's most of the reason why cancer has become the number 2 killer, because of the fact that once we catch it, most of the time, it is too late. And it's not at the very, very beginning, like this Galleri test gives us a leg up on.
Right. So I would say it's not necessarily when they're invisible. It's just when they're really small. So
that's to the naked eye, I mean, when I say that.
Yeah. So when you this can potentially start picking stuff up before it is what we would say clinically relevant, because as we said before, some of these cancers will potentially resolve on their own. Right. But remember, you mentioned earlier that there's only 5 screening tests.
Yeah.
So if we're not screening for something, and even if it's just stage 1 and we can see it on imaging, how often do you just routinely get imaging? You don't, right? I mean,
right.
So we talk about pancreatic cancer, for example. Pancreatic cancer, we use the example again, and I'll say it again, and everybody knows a story about some healthy person who all of a sudden got pancreatic cancer and died like 3 months later.
Mm-hmm.
And it's not because pancreatic cancer itself is inherently so much more aggressive than every other cancer. It's just that we don't find out about that cancer until it's advanced, because we don't screen for it.
Yeah, we don't have
a screening test for it. So if we can identify these cancers in stage 1, and they are visible, we're just not looking for them, right? Then we can treat them. Let's just put this in perspective. So cancer is our number 2 cause of death, right? Cardiovascular disease at this point should probably not even be in the top 10, because of our ability to not only deal with it early, but even if it is a problem, to treat it aggressively. On the other hand, if you've got cancer, it's a completely different game. We don't have the tools that we have in cardiovascular disease to treat it. Cancer, in my opinion, is going to very soon become the number 1 cause of death, because cardiovascular disease, we just have so many tools at our disposal.
Yeah. Well, also, if we don't start doing these Galleri tests, or people don't start doing these, I mean, just being educated at the fact that we only have 5 tests that really show cancers at an early age, that's tests that are common, then it, like you said, it's just going to exponentially get worse and worse, because we don't really have that.
Yeah, and we have to remember the big thing here is that since we don't have great ways of treating advanced cancer, the best chance we have is either, avoiding cancer in the first place, which is not easy always, or finding it really early. And so the early detection is really the best offense at our disposal that we possibly have against this whole disease process. So I'm a big advocate for this kind of early screening. We're also going to talk about full body MRIs too, which is another way you could do it.
Something that stuck out in me in the interview too was a lot of the misdiagnosing, and that's because we don't have these types of tests, and we're so limited on the ones that we do actually have.
The one last thing I would say is that there are doctors who don't really like this test, and their argument is we're getting a bunch of these. We're getting a lot of these tests that are saying that there's cancer, and we can't see any cancer. And so
that's so idiotic in my opinion. That's so, like, some doctors, and I mean, I'm going to just speak really frank. Like, some doctors just have this like ego, or this like godlike complex. And so if they can't see it, or they can't understand it, or they're not the one to discover it, they get, I don't know, whether it's, like we're saying with AI, some of us get really scared about some of our jobs. Like, maybe that could be a deep down subconscious issue where they just are like, well, that's dumb and they're going to take the stance against it.
Yeah, I think this is God speaking, by the way. I think the argument is a little bit more nuanced than that. I think they're concerned about, false positives, things that are going to potentially be what we would call subclinical.
But they have a bunch more false positives.
Yeah. Yeah, no, I get it. But I think I'm just putting that argument out there because certainly there's going to be some physicians who probably have experience with this are going to be like, well, you're not understanding.
And I'm putting that argument out there, so that's my side.
Yeah. Okay. Well, good.
Well, I'm going to read us out. Longevity Junky is created by Dr. Buck Joffrey, MD, and Timothy Fonara. It is executive produced by myself, Nikki Lee, Buck Joffrey, and Timothy Fonara. The show is produced by Pretty Fast, and Phil Ferragamo is our producer, and original music by Alloy Tracks. We're distributed by our friends at Acast, and see you next time.